Okay, I think we can start. I will just ask all of the participants to mute themselves while the talk is going on and the question and answer session. Agnese, your turn.
All right, so welcome everyone to this journal club on this paper that was published in PRL recently. The title is State-of-the-Art Estimation of Protein Model Accuracy Using AlphaFold by James Roney and Sergey Ovchinnikov. The moderator for today's journal club is
if I'm still testing
Roney.
positive, then I would not return.
Sorry, there's someone whose voice I can hear. If you can just all mute yourselves. Thank you. So as I was saying, the moderator today is Dr. Bodhi Vani. Bodhi did her undergrad at IIT Bombay in India. Then she moved to the University of Chicago, where she did her PhD under the supervision of Aaron Dinner and Jonathan Weir on the trajectory stratification for enhanced sampling methods. And now she's currently a postdoc at the University of Maryland under the supervision of Professor Pratush Tiwari. And she's interested in machine learning and enhanced sampling of proteins to solve biological problems, specifically structure and dynamics-based drug design. So before we begin with Bodhi introducing the speakers for today, I just wanted to go over a few slides. So just to remind you a little bit of the history of PRL, where this paper was published. PRL has been publishing top research in physics, as it's shown by this slide, where we collected papers that have been published in PRL and have been mentioned by the Nobel Prize Academy for the past 12 years. So we were very happy this year in 2022 that we could, add one more year. And we hope that this trend will continue and that we will keep getting, the best papers in the physics research world. Just a quick reminder, please mute yourself, but unmute yourselves at the end of the presentation if you want to ask questions. I will ask you to please refrain from asking questions during.
Oh, sorry, Agnes, I accidentally muted you. Could you unmute yourself? Oh, Agnes, sorry. Looks like you got muted by accident.
Can you hear me now?
Yes.
Oh, sorry. I think someone muted everyone. So, okay. Please unmute yourself if you want to ask questions, but wait until the end of the presentation to do so. You can raise your hands or type them in the chat, and someone will read them out for you if you don't want to do it yourself. But please feel free to unmute yourself and raise your hand and ask questions. And the last thing that I wanted to mention before we start is that because this talk is about proteins, this is the right audience to make this brief announcement. APS is launching a new journal, PRX Life. It will be a highly selective and open access. You can Google the website is on, so you can just Google this web page and read more about it. It will be open for submissions in 2023, so beginning of January, and the fees for publication will be paid for the entire year until 2024. So with this, I will stop sharing my screen and I'll pass it on to Bodhi for introducing the speakers.
Hi, Yeah, so I'm really excited for today's talk about the paper State-of-the-Art Estimation of Protein Model Accuracy Using AlphaFold, which is a really interesting piece of work discussing and actually answering questions that I think a lot of us have been asking ever since the paradigm shift that AlphaFold brought to the field. So introducing the authors, James Roney is a recent graduate from Harvard with a bachelor's in computer science and a master's in statistics, with a prolific undergraduate career winning several awards, including a prize for outstanding senior thesis. Apart from protein folding, he has worked on continuous time Markov processes, as well as work on data security and data structures. He's now a machine learning research engineer at DSHA Research. We also welcome Dr. Sergey Ovchinnikov. He is a John Harvard Distinguished Science Fellow at Harvard University. His independent research group works on understanding protein folding and evolution using machine learning techniques in combination with protein sequence data. His group also brought us CoLabFold, which made AlphaFold accessible to everyone. Before this, he did his PhD at the University of Washington, working with David Baker on protein structure determination using evolutionary data. So without further ado, James, if you want to start the presentation.
Yeah, great. Thank you so much. It's really great to be here talking today. And yeah, thank you all for your attention and for coming. And thanks for the great introduction. So I hope everyone can see my screen all right. Does that look good? So then I'll go ahead and start presenting. Okay, yeah. So hi, my name is James Roney, and today I'm going to be presenting on our work on our recent paper in PRL called State-of-the-Art Estimation of Protein Model Accuracy Using AlphaFold. And this work was work that I did while I was at Harvard, and along with Sergey, who is here on the call today, and will be answering questions and speaking a little bit later. Okay, great. So I think I'll start by giving a sort of brief outline of what I'm going to talk about today. So I'll start by giving some background on the protein folding problem, and then we'll talk about AlphaFold, and some of the specific contributions of this paper, which involved decoy scoring, our specific findings, and then some sort of conclusions and perspective. Okay, to start with background on protein folding, as many of you know, proteins are like the macromolecular workhorses of life, and proteins, their function is really dictated by their 3D structure. And so determining 3D structures has been, sort of a staple of structural biology for decades. And understanding the 3D structures of proteins is important for building just sort of a mechanistic understanding of how molecular biology works, and also for things like designing drugs and lots of other things. And so, historically and still, the major tools we use for determining structures are like cryo-electron microscopy and X-ray crystallography. But for a long time, we've known that proteins' 3D structures are actually determined by their linear amino acid sequences. We've known that since Ann Finson's like seminal refolding experiments. So there's really been this sort of idea, like, what if we could predict the 3D structure directly from the amino acid sequence? And this has been thought to be possible for a long time, and people have been working on it for a long time. And it has the potential to sort of save a lot of experimental effort in protein structure determination. So speaking a little bit more to the thing I mentioned about how protein sequences determine structures, we know that proteins in theory fold to minimize free energy, which is just going to be a function of the protein sequence and sort of the conformation that it adopts in terms of sort of its backbone and side chain angles. And if we sort of understand the free energy landscape of how a protein folds well enough, then in theory, we should be able to predict the protein structure just from that primary sequence alone. So now I'll talk about some of the more recent progress in protein structure prediction, specifically that's been ushered in by AlphaFold within the last few years. So AlphaFold has demonstrated some really impressive performance predicting protein structures. And basically the way it does that is using some ideas that have been sort of in the field for a while now. So basically the key ingredients to AlphaFold structure prediction are sort of a target amino acid sequence that we want to predict, and also a multiple sequence alignment. So the multiple sequence alignment is basically a collection of protein sequences that are evolutionarily related to the target sequence. And you can imagine them as coming from different species, like, this human, this fish, and this rabbit. And what's really cool about this is that if we look inside the multiple sequence alignment and see sort of multiple positions that are linearly distant in the amino acid sequence, but we see sort of correlated patterns of mutations in the multiple sequence alignment data, that can actually give us an idea of where there are structural contacts inside the protein. So more specifically, if we see sort of two columns within this multiple sequence alignment, or we see sort of mutations happening in tandem, what that can signal is that if you have two residues that are sort of in contact, when one of them mutates, that can sort of induce a selective pressure for the other to mutate. And so we see that as these sort of covarying mutational patterns. And for a very crude analysis, you can just literally look at covariance patterns and see these bright spots that could indicate possible contacts in the folded protein structure. And so that sort of evolutionary signal has been really, really important for recent developments in protein structure prediction, including AlphaFold, but also, several generations of models that came before it using things like Markov random fields. And so the really important thing about AlphaFold is just to combine sort of this idea of protein structure, of sort of using evolutionary information to determine protein structure with sort of deep learning techniques and a lot of careful engineering to produce really good protein structure predictions. And then some other cool inputs to AlphaFold that we'll talk about that will become important later are sort of these template inputs. And that's basically the reason that exists is so that the model can sort of look at previously determined protein structures that have sequences that are homologous to the target sequence. So for instance, if there's already been a structure solved that's in the same protein family as the sequence we're trying to predict, then that is sort of useful information that the model will want to know about. And this is sort of analogous to classical homology modeling for protein structure determination. Okay, so I was previously talking about the importance of a multiple sequence alignment for AlphaFold structure predictions. And while it's technically possible to produce predictions from AlphaFold without using a multiple sequence alignment, in that case, the prediction quality deteriorates quite a lot. So this plot here shows that if we have a multiple sequence alignment, AlphaFold generates very high quality protein structure predictions. This is a metric called template modeling score that ranges from 0 to 1, 1 being very good, and below 0.5 generally being of pretty low quality. Whereas from a single sequence, AlphaFold doesn't do very well. So this sort of presents kind of a puzzle, right? So we talked about how multiple sequence alignments are informative for determining protein structures. But if we go back a few slides, we said that in theory, this free energy landscape should sort of dictate the native structure of a protein. And so, if this Amphenson's dogma is true, then it seems like, well, why can't we just sort of learn this energy function? Why do we need the multiple sequence alignment information? If in theory, we could just learn to understand the conformational landscape, and then determine the minimum energy native structure. So if the sequence contains all the information we need, then why are our methods still so reliant on coevolution data from multiple sequence alignments? Why can't we just learn to minimize that energy landscape? And so we were thinking about this and we came up with a possible explanation, which is that while it's theoretically possible to predict protein structures by minimizing the sort of potential energy function, this is a really difficult global optimization problem. Because if you just think about the number of conformations that a protein can adopt, there are really an astronomical number. And so, and this is related to something called Levinthal's paradox. And even if, thankfully, we know that you probably don't need to search all of those conformations because hopefully we hope that the energy landscape is like relatively smooth and sort of funnel shaped, so you can find your way down to a minimum. But I mean, still, that could be a lot of a big sampling problem. And we've seen this before with molecular dynamic simulations of protein folding, as well as simulations using energy functions like Rosetta. It seemed like, you know, there's really this sampling bottleneck where sampling enough structures to find ones that truly have low energy and are stable is a really difficult sampling problem. So we wondered, what if it's the case that AlphaFold has learned sort of a potential function for assessing the accuracy of candidate protein structures, but it needs the MSA to find a good starting point for globally optimizing those low energy structures. And so to give a sort of graphical illustration of this hypothesis, what we sort of hypothesized that maybe AlphaFold has this sort of learned energy function where if you give it a candidate protein structure, it can say, yes, this is sort of a good stable native structure for the sequence, or no, I think this is a high energy unstable structure. But given sort of AlphaFold's, you know, limited ability to do the structure search, what it needs to do is then look at this multiple sequence alignment to sort of give itself a good initial guess and locate sort of a coarse-grained global view of what the structure generally looks like. And then perform sort of a local optimization in that sort of, using that sort of learned energy function. And also another sort of output of AlphaFold is how confident it is in its predictions. And so we hypothesized that within this framework, then this sort of internal potential might be reflected in the confidence values. So this is kind of a neat story, and it would explain why AlphaFold remains reliant on MSAs, even though it can achieve such high accuracy when it does have that coevolution information. But you know, how exactly would you test something like this? And that's sort of what we set out to do in our recent work, was to test this hypothesis. So basically, and so we went about that hypothesis, testing that hypothesis using something called decoy scoring. So given our hypothesis, we don't really expect AlphaFold to be able to predict accurate structures without MSAs. So here's an example of that. When we get rid of an MSA, AlphaFold really is struggling to predict the structure accurately. You know, it has this TM score of 0.46, which isn't very good, and it's also not confident. But we thought, well, if AlphaFold has potentially learned like an energy function to assess the accuracy of candidate protein structures, then what if we can insert sort of a candidate protein structure here via AlphaFold's template mechanism for learning about homologous structures? So we reasoned, okay, what if we supply a decoy structure to AlphaFold that's sort of acting as like a candidate structure, and we're sort of querying the model to ask, how accurate do you think this structure is? And then from the AlphaFold output, we then look at these confidence metrics, and we wondered, well, can we use these confidence metrics to accurately estimate how accurate the input decoy structure was? And specifically, we did that by computing what we call the composite confidence score, which are basically, we took the product of some of AlphaFold's confidence outputs, and also we multiplied that by sort of the TM score difference between the input structure and the output structure, just to reflect the fact that if AlphaFold's prediction differs a lot from the structure we gave it as a candidate, then that probably means that it's not very confident in that input, or that I guess the output confidence structure might not be very indicative of the quality of the input structure. So the fundamental question we're asking is whether AlphaFold's confidence is a good predictor of the quality of these decoy structures. In other words, can AlphaFold recognize a good structure when it sees one, even if it can't predict that structure without access to coevolution information? And we found that the answer was yes. So even though AlphaFold can't predict protein structures from scratch, its confidence signal does a really good job ranking candidate structures, even with no MSA. So here we have an example for one protein target where we have a bunch of candidate structures called a bunch of candidate decoy structures that we supplied to the model. And then we computed this composite confidence score. And we see that there's this really nice correlation. Even though AlphaFold struggled to predict the structure when we didn't give it any sort of template input and we didn't give it any MSA, this red line represents the quality of AlphaFold structure prediction. without any template inputs. So AlphaFold can only predict structures of like quality under 0.5 in terms of TM score. Yet when we give it high quality decoy structures, it's readily able to recognize that those are high quality decoys and produces outputs that are nearly perfect with very high confidence. So this is interesting evidence that AlphaFold, even without co-evolutionary information, is able to distinguish and score the compatibility between a protein sequence and a protein structure, even though it can't predict that structure from scratch. And so to get into more specific numerical findings, we find that on this protein structure ranking task, AlphaFold outperforms other sort of energy functions that have been developed in the past, including classical statistical potentials like Rosetta, and also deep learning based architectures for protein model accuracy estimation like Deep Accuracy Net. And just reiterating, AlphaFold also does a really good job ranking, even though on this set of in this set of data, AlphaFold does a really poor job predicting protein structures in the absence of co-evolutionary information. It can still do a really, really good job at this ranking task in the absence of co-evolutionary information. Yeah, so here's just another scatter plot indicating how on this particular data set, AlphaFold a strong majority of the time outperforms other previous potentials that have been developed for this challenge. And we also tested on a novel set of proteins from the CASP14 data set. And we found that, again, AlphaFold did a really good job estimating the accuracy of protein models, better than previous energy functions, other deep learning methods, and also even some that you made use of multiple sequence alignments. So I think this is just further evidence that, even though AlphaFold can't predict structures very well without access to co-evolutionary information, it can really, it still does a great job at recognizing high quality candidate structures, even in the absence of any co-evolutionary input. So another interesting thing that we found is that when we sort of supply a structure as a decoy, the output structure from AlphaFold is almost always higher in quality when measured by something like TM score. So this also supports the hypothesis that AlphaFold sort of derives starting hypotheses from co-evolutionary information or from templates, and then uses this sort of learned energy function for local structure refinement. And we see that reflected in the fact that accuracy gets improved as we pass these decoy structures through the model. This is sort of a technical detail, but one thing, when we in our initial exploration, we sort of found that if our decoy structures are associated with the sequence, it's actually sort of the native sequence of the protein structure we're trying to, each decoy structure has sort of an amino acid sequence associated with it. And if that sequence matches the target sequence that we're trying to predict, actually AlphaFold is very confident all of the time. And this sort of makes sense because in its training regime, if you see a template that has very, very high sequence identity to the sequence you're trying to predict, that's probably a pretty sure bet that that template is more or less correct. And so the model just sort of inherently is highly confident. But we found that we could get this really interesting ranking behavior if we just sort of replace that sequence with a sequence of gap tokens. So that's what we did in all of these experiments. So just to wrap up here, the key takeaways are that AlphaFold can score candidate protein structures very effectively, even without an MSA, even though it can't predict those protein structures from scratch without an MSA. And this supports the idea that AlphaFold has learned a sort of internal energy function, but needs those MSA and template inputs as a starting point for sort of searching for an optimal structure. And this idea has subsequently been supported in the design of protein-protein interactions. So this is from the Baker group, and they found that sort of they used a similar trick to score protein complex interfaces. Then the sort of confidence that AlphaFold gave as an output was very highly correlated with sort of the experimental success of designing those protein-protein interfaces and having them remain stable. And then another recent structure prediction network called RGN2 has also made use of this work to effectively refine the structure predictions that they made using sort of the same AlphaFold template mechanism. And so I think there are some important further questions raised by this work. So I think we've shown that AlphaFold has this sort of energy function that does a good job scoring the correspondence between structure and sequence without the need for coevolution information. I think, there's still a big question of to what degree this reflects protein physics. So, it seems likely that there's sort of some idea of basic principles like how hydrophobic amino acids should be concentrated towards the core of the protein, and what favorable hydrogen bonding looks like. But then again, there's probably some departure. I wouldn't expect it to sort of adhere to all of the contours of an actual, truly accurate physical free energy function. So like, to what degree does this AlphaFold confidence energy function truly line up with what we expect based on physical calculations? Some other questions are how sort of AlphaFold single sequence decoy discrimination abilities compare to the abilities of other recent efforts at sort of reducing the dependence of protein structure prediction on MSAs. So these are mostly methods that use language models to sort of circumvent that issue. Another issue is how we can deal with sort of if we want to sort of ease the dependence of our methods on coevolution information. It might be more difficult to deal with sequences that sort of oligomerize, so they sort of interface with multiple copies of themselves or other proteins. That's sort of a signal that we can sometimes dig out of multiple sequence alignments, but it might be more difficult if we don't have that information. And yeah, I guess a final question is what's the best way to sort of take these results and apply them to get better structure predictions, whether that means doing that in cases where we have limited MSA information, or just on protein examples that we have where our methods have limited success. And so in terms of the applications, I think Sergey has some additional slides and is happy to answer questions on that as well. But I think I'll end my presentation here, and and I'm happy to answer questions.
So yeah, feel free to ask questions in the chat or raise hands and unmute yourselves. That was an amazing talk. Thank you so much. Also a very cool paper. I think Arthur has a question. If he wants to unmute himself and ask, that's good. Otherwise, I'm happy to read it out. Okay, have you tried deriving a mathematical form of the energy function, maybe by making small perturbative changes to the decoy structures and checking the change in confidence score, kind of like taking a Taylor expansion?
Oh, that's an interesting idea. We have not, but that would be interesting. I mean, I think a Taylor expansion could be interesting. I mean, like classically from physics, we know that anything locally can be a harmonic potential, right? So we can probably create some local approximation that way. My concern is that AlphaFold is an extremely complicated, model with like hundreds of millions of parameters. So my guess is that the analytical form of this function is going to be not super amenable to coarse graining, but it might be. I mean, I think this is sort of a fundamental challenge in modeling protein physics, right? I mean, our sort of classical molecular mechanics, force fields, and also things like Rosetta have the benefit of having very interpretable, simple functional forms. But there's sort of been the success of using them for protein structure prediction has been more limited. And as we show in this paper, even for the ranking task, AlphaFold also does better than those more classical methods. So I think there's probably going to be a trade-off between how interpretable the functional form is, and sort of how accurate it's going to be.
Zach has raised his hand, I think, immediately after that question. Zach, if you want to unmute yourself.
Yeah, sure. Thank you. I really enjoyed the talk and the paper. So in our use of AlphaFold, we've seen that it really struggles with point mutations that, for instance, might swap the ranking of the active or inactive structure. Have you seen any evidence in your testing of your method that it could do better with point mutations? Because that would have really exciting implications for disease.
Yeah, I mean, I think it's sort of been known that AlphaFold doesn't do super well with point mutations. I think there are some reasons for that. I mean, ultimately, AlphaFold probably was mostly trained on stable structures, so it's sort of its ability to sort of understand the relative importance of, it's probably more attuned to more global features rather than like these small perturbations to one residue or another. We have done some testing to see whether sort of this method. So basically, we were mostly focused on the problem of ranking structural differences given a sequence, but there's sort of this flip problem of ranking sequences given a structure, right? And we've done some work on that. I think Sergey has some additional slides if we want to touch on that briefly. What we found is that it can work, but I think there's some more investigation needed to determine exactly how well it works. I mean, I think in terms of, yeah, I mean, we've seen that it can work and can work sort of like, I think in sort of the ML sort of universe of predicting mutational effects, a lot of the work has been with sort of evolution-based models like language models, and also just, you know, you can use Markov random fields over multiple sequence alignments to do that sort of thing too. And so yeah, so it'd be interesting to know if you could sort of do that even without, based on structure, but without necessarily needing to leverage as much evolutionary information. And we found that we've seen promising results, I think, but not quite as strong as the results the other way around in terms of the structure ranking. Yeah, and I think we probably have some data on that if we want to show it.
All right, thank you very much. That was really, really cool.
Eric Chortle, I'm sorry if I'm mispronouncing your name. Can you elaborate more on the gap token?
On what?
The gap token.
Oh, yeah. So basically, when AlphaFold, this is just sort of a technical trick, but what happens with AlphaFold is it takes sort of this, it takes the MSA, the target sequence, and sort of this template information. And the template is basically designed to be a homologous, basically to be an input that represents a homologous protein structure, right? And so it has structural features, and it also has a sequence associated with it, a sequence that generally is completely different from the target sequence, right? So what we're doing when we try to feed a decoy structure to AlphaFold is we actually want to feed in a structure that has the exact same sequence as the target sequence, right? Just a sort of a made up structure. And the details of how these decoy structures are generated is widely varied, but basically the way we've done it is we've used data sets that are basically composed of protein structure predictions that use various methods and that are of various accuracies, right? So naively, the thing you would do is just give it this this decoy structure, and the sequence attached to the decoy structure you would just set to the target sequence, because that really is the sequence of the structure you're passing in. Unfortunately, what we found is that AlphaFold then is always very, very confident in the output. And that kind of makes sense, right? Because if you give the model a template sequence that has the exact same sequence as the structure as the sequence is trying to predict, it'll just say, oh, well, you know, I already have the answer, so I'll just be very confident in my output. So that was kind of disappointing. So we just said, well, what if instead we just sort of replace replace that sequence, that template sequence with a sequence of gap tokens. And these gaps are just sort of these characters that are meant to represent missing amino acids. And we found that by doing that, we got this to work very well. But really, it's just like a technical thing. And these experiments haven't been shown here, but we've also found that if we can, there are many other ways to circumvent this issue. If you for instance, want to fine tune the model on a little bit of extra data to make it so that it doesn't suffer from this overconfidence issue, you can also do that. So it's just sort of a way to get around some quirks of the way the model is kind of wired.
Namrata, would you like to unmute yourself and ask your question?
Yes, thank you so much. It's a wonderful talk. I'm just wondering, is there any way that you could get insight into the secondary structures forming? Like, is there any way that you could probe as the protein is folding in order to get some idea of the order of secondary structures being formed at all?
Oh yeah, that's an interesting thought. I mean, do you mean in terms of the actual pathway that the protein would fold in an actual cell? Yeah, I mean, I think I would interpret any of that sort of with some caution, because I think ultimately what we've shown is that you can do good structure discrimination when you're actually at the native structure. In terms of intermediate structures, I feel like we know less in terms of what actually, in terms of what this confidence score would actually be indicating. Yeah, but there are potentially some ideas. I mean, yeah, I mean, I don't think we've really thought about that, but I think there are plenty of things you could do. You could try doing some sort of I don't know, if you wanted to sort of elucidate the folding trajectory, maybe you could try to use some sort of molecular dynamics approach, where you sort of create a biasing potential using AlphaFold in some way. But yeah, I think that's a tricky problem. And I mean, another thing we've done is we've tried to sort of use this approach to predict protein structures from scratch, where we basically just sort of use the AlphaFold energy function as sort of perform gradient descent in structure space, kind of. And we found that we can use that to improve protein structure predictions over AlphaFold's sort of naive single sequence predictions. I'm hesitant to say that the trajectories we see there actually represent real folding pathways, because I think fundamentally that's not something we know yet.
Right.
But I think there are some exciting possibilities. Yeah.
When you said you did that, did the data you got, was it time evolution data? Were you looking at it in snapshots at all? Or
yeah,
is it more
we have some time evolution data and some animations that Sergey has made that we could maybe show. But like I said, so we can perform descent in this energy function and get better structure outputs. Like I said, I am somewhat hesitant to say that these are what real folding trajectories look like, though.
Can I just say one more thing?
Yeah.
I think one of the reasons I was asking that is looking into misfolded proteins. So did you ever run across something where you used a particular decoy structure, and then the prediction that you got was similar to other misfolded proteins that we know exist? Like, for example, when someone has Alzheimer's or something, we know that it's an aggregate of misfolded proteins. So is there a way to predict a misfolded protein, which is still it's in a minimal energy?
Yeah, that's a really interesting thought. I don't think we've looked into that, but I mean, if you have if there's a canonical example of proteins that have a native structure and then a misfold, right, it would be interesting to see sort of how the energy minima would play out there. That's also kind of an interesting case, because I guess it's a case where you might expect coevolution to not work as well, because maybe the the misfolded state isn't really influencing the evolution of the protein family as much, because it's kind of already existing in that state at all is kind of already deleterious.
So it's possible that your low confidence score for that particular one still is a misfolded protein, but it gives you a low confidence number because it's not this like, I don't know, it's not the right predicted structure. So
I'm not sure what it would look like. I think it would be really interesting to try. I mean, I think, you know, it would be interesting to see if sort of we see, I mean, yeah, I guess a really another important sort of test that we've done some preliminary work with, but that we haven't investigated in as much detail is looking at sort of protein energy landscape where we know we have multiple minima, and seeing if those minima are sort of being mapped appropriately. But doing that with a misfolded protein seems like a really interesting and great direction. So that's something that I think would be really cool.
It was just something interesting.
Oh,
I will email you because I don't want to take up more time.
Oh yeah, no problem. Yeah, please do.
Thank you.
Akash asks, PLDDD score is interesting. I might be wrong, but isn't PLDDD score being calculated in the structure module of AlphaFold 2? If that's the case, then can you just do this similar experiment just for the structure module alone with perturbing the pair residue matrix?
Yeah, the PLDDT is computed in the structure module. Can we do this just in the structure module alone? My guess would be no, because there's a lot of sort of implicit internal representations that go into the structure module that sort of, I would imagine, have to come out of the previous layers of the model in order to really make any sense. But if I'm misunderstanding, please correct me.
Akash, if that was that. Okay, he'll ask a follow-up if that isn't the question he was asking. Terry Bollinger asks, related to the perturbative idea mentioned earlier, could you implement a secondary learning system to analyze perturbed AlphaFold results to discern what variations lead to high MSA-like scores?
Sorry, could you repeat that?
Related to the perturbative idea mentioned earlier, could you implement a secondary learning system to analyze perturbed AlphaFold results to discern what variations lead to high MSA-like scores? So like augmenting two channels, I think is.
I can ask that directly if you want.
Yeah, sure, go ahead.
It's just what fascinates me about your system is it has learned something that it doesn't seem to understand what it's learned. It's like it's learned how to ride a bicycle, but it doesn't know how it rides a bicycle. It can't say how it's able to do that. And I'm just intrigued that if you could apply a secondary learning system to look at those, perturb those results, as someone mentioned earlier, it seems like the secondary system might be able to say, oh, here's the variations in the sequences that wind up not making a difference or doing to them. It seems like your AlphaFold itself has information that could be learned by another system. And
yeah, sure, yeah. I guess this is sort of related to the idea of like knowledge distillation in machine learning sort of parlance. And I think that's something that we're really interested in because one thing is that, you know, I think that sort of distillation can be helpful both in terms of interpretability and also in terms of like performance, just because for doing what we're doing here, I think we probably don't need the full, you know, 300 million parameter or whatever AlphaFold model. We could probably distill it into something a lot smaller and something that might be easier to run if we want to sort of, I don't know, score a bunch of dynamical trajectories or try to perform more of this sort of coordinate descent in structure space, or if we want to do more sort of computationally intensive operations. And you're right, I think it also would be good for probably for interpretability purposes in terms of understanding sort of what the actual determinants of these model confidence scores are.
Thanks.
Hugo Bohorquez has a comment. I'm not sure what the question is in there, but if you have any like comments on what he says. He says the secondary structure of proteins is highly correlated with Dayhoff's matrices, which are the bioinformatics way to address amino acids evolution. It would be interesting to see how that information is projected inside AlphaFold.
Yeah, I agree. I mean, I think, yeah, I mean, I imagine that, you know, secondary structure determination is probably a significantly easier problem than determining the tertiary fold. But yeah, I think that, you know, I'm sure that if you were to probe more closely, you would find, my hunch is that if you were to, you know, probe more closely, you would find that sort of some of these classical ways of determining things like secondary structure are probably correlated with sort of the energy scores that we're getting from AlphaFold for those particular things. And, you know, I think, yeah, but I think, yeah, so I think that's a good point.
If there are no other questions, I'll jump in and ask a question.
Go ahead, please.
So one of the, like, there are a lot of other algorithms that have been coming about, like ESM fold and
yeah,
and they don't use MSA. And I'm wondering how do they kind of solve the localization problem? Like, how do they sort of seed the optimization to make it?
Yeah, it's really interesting. I mean, I think, I mean, there might be people here who disagree with me, but I think that it seems pretty much like, so these models all use language models, right? So they've been trained on a ton of unlabeled protein sequences. So I think the hypothesis is that they can just sort of reach into their memory banks and pull out something that's very close to an MSA just by recalling all of the unlabeled sequences that they've seen. And I think there's some pretty strong evidence for this. So if you look at these protein language models that have been trained on a bunch of unlabeled protein sequences, and then you look internally at them and look at sort of how like the attention is propagated between residues, you can actually see like contacts inside those attention maps. And that's just simply because, you know, like we were talking about before, if two positions in a sequence are sort of co-informative about each other, that's sort of important information in terms of contacts. And what these models are actually trained to do is to predict masked out tokens, right? So that sort of inherently gives them this inductive bias towards looking for other positions in the sequence that are informative about those tokens, right? So if you think of a big transformer model that's been trained on a bunch of protein sequences as really just sort of a bunch of compressed like POTS models that have been trained on individual MSAs, then this starts to look it starts to look less like an MSA free prediction and more like sort of an implicit MSA prediction. And the evidence for that, I think, comes from the fact that on proteins where if you do compile an MSA, that MSA is shallow, the language model structure predictions are much poorer. So yeah, I think a real test, and I think this is something that we're still as a community are sort of trying to figure out what the best test is, would be to really just find a set of proteins that are difficult to predict based on evolutionary information, and where, you know, if we have a model that all of a sudden is doing really well on those, it sort of indicates that we have something here that's going beyond what can be found from evolutionary analysis alone. This paper that I referenced towards the end of the talk called RGN2 did that for a small set of proteins, but I think, and they actually, and they made use of our approach in sort of predicting these so-called orphan proteins. But I think the consensus is that in terms of creating evaluations like that are really testing the ability of protein structure prediction to be independent from evolutionary information, we have sort of a long way to go towards figuring out what the right way to evaluate that is.
Since there are no more questions, I will jump in and ask a follow-up question.
Yeah, go ahead.
So if more people have questions, please feel free to either write them in the chat or raise your hands. So it seems like that implies that they've trained something that does something similar to MSA. I'm wondering if there is a principled way to so like one of the things that we've been doing, that a lot of people have been doing, is just like decrease MSA depth and then like try to learn something from the output. And I'm wondering if there's a more principled way of doing this, either by like pushing around the things that these other language models have learned that implicitly generate something MSA like, where we can learn either misfolded states like Namrata was asking, or just more energy minima like configurations that are not native like but close to native like in stability. Like, is there a more principled way to seed the search essentially?
Yeah, I mean, I think that's a good question. I mean, I don't know if language models make that easier. I mean, in some sense, the sort of MSA clustering approach for AlphaFold that you mentioned doesn't really work at all, although I'm sure Sergey and other people have figured out some hacks to make it work. I feel like I've seen that on Twitter somewhere. But I mean, there's always some hack, but I don't know of any way that language models make that easier. One thing is, I don't know, like, I mean, I think there's interest in making protein structure prediction more probabilistic, inherently probabilistic. I mean, we've seen this flurry of protein diffusion model activity recently that maybe could aid with something like that. And also, I don't know, our sort of idea of using AlphaFold to create a sort of energy function, sort of in theory at least, sort of opens the door to some alternative conformation sampling. But as we've talked about before, the sampling problem is hard. So I think I would be interested in seeing new research that sort of tries to use machine learning techniques to tackle the sampling problem, which is something that I feel like I've seen in other domains, like where I guess you just have a hard sampling problem that's difficult to do via MCMC or molecular dynamics. Trying to use some sort of generative type model to overcome that, or I think would be an interesting direction for sampling all kinds of different protein related problems. But yeah.
Thank you. Those are very interesting directions. Terry, would you like to unmute yourself and ask?
Yes. This is just a broad question, but I'm curious. You presented a lot of really fascinating aspects about how you get the same folds from these analog sequences. What is the level of the physics understanding of why this is happening right now? Has there been progress in which people have a deeper understanding about why this phenomenon even occurs? What's the state? And I realize that's a kind of an unfair question because you're trying to get at with the modeling, but what's going on in the physics understanding side of this to really get a deeper grasp of why these things are happening?
Yeah, I mean, that's a good question and a hard one, but yeah, I mean, I would say that right now, I mean, this was our sort of initial probe into understanding, well, what can AlphaFold do in the is AlphaFold really just doing bioinformatics on MSAs really well, or is there something to it other than that allows us to sort of assess the correspondence between sequence and structure? And so I think we've sort of answered yes on that question. And then determining exactly what the determinants are and how close they are to sort of a physical energy function is a tough problem, and one that I think we're just sort of scratching the surface of. I mean, I think there were some good ideas talked about today in terms of doing perturbations, looking at secondary structure, looking at alternative minima to figure out what the determinants are. But yeah, I mean, I think there are a lot more steps to be taken in terms of physical interpretability for sure.
And you've been saying this, but I would just agree with it that AlphaFold seems to have captured some of that data in a fashion that may be tractable for a direct analysis from there. And that is what intrigues me most about your talk. Is
Yeah. I mean,
you wouldn't expect.
What'd you say?
It's doing things that you wouldn't expect it to be doing, and that's fascinating.
Yeah, I think so too. Yeah, so I think there's a lot of interesting work here in understanding even further, right? Like, yeah.
Thanks.
Do we have time for another question, Agnesa and Stohan? All right, yeah, Namrata, go ahead.
Thank you so much. I have a quick question, actually similar to what Terry was saying, that what kind of data do you, on energy landscapes, do you have that we could download and then run calculations? Like, do you have that at all, available for everyone?
On energy landscapes, well, we have all the data from the paper public. So all of every structure that we analyzed in the paper is published. So that's all. And then we also have we have Colab notebooks where you can run this method as much as you want if you have your own energy landscape data that you want to explore.
Okay. No, that's good. Thank you.
Yeah.
All right, thank you so much, James and Sergey. And this was a very cool discussion. I will hand it over to Agnesa to conclude.
Yeah, thank you so much, Bodhi, for moderating the session. Thank you so much to James and Sergey for being here and presenting. We are right on time, 2 minutes early, so that's great. And thanks to everyone who participated, asking questions and just listening in. And keep an eye on for future journal clubs that we have. We try to have every month. So just keep it under your radar, and we will send information for the future ones. And thanks again to everyone who participated. And thank you, Stoyan, for organizing everything as usual.
Yeah, thank you very much.
Yeah, thank you so much, everyone.